Hello everyone, I welcome you back to another important lecture where we would be discussing the topical past paper for this chapter number 4 that you have for your IGCSE's Biology, and this goes with the name of the biological molecules. Among the biological molecules, you may have studied about the carbohydrate, the protein, the fats, and the nucleic acids, and now we would see in this video that how come different past papers they have come over the year from the year 2013 and then coming till 2025, and we will try to understand the dimensions of it. If I begin with question number one, so this happened to appear in the May-June of 2013, and at that time the variants were not as 4142 as we have now. So this was variant 31 with us. And here we have got some of the students, and they are investigating the digestion of the fat that we have in the milk. Now, if I were to quote you some of the facts out of the fats, what we know as that if I would take total of three fatty acid chains and when I would combine them with one molecule of glycerol. So as a consequence of that, we would be making a fat molecule and there would be removal of three molecules of the water. And in the chapter for the nutrition in humans, the teachers may have taught you that if we are eating the fat and they had to be digested in our body, then there would be two mechanisms involved in the fat digestion. One of the phenomenon that takes place in the fat digestion is emulsification, where the large fat droplets that we have with us, they would be physically being broken down into the small fat droplets, and this is happening due to the bile salts. And following this, it would be acted upon by the enzyme that we have with us. This is a lipase enzyme, and it would break down the small fat droplets into the fatty acids and glycerol. Now, after we have broken down the fats into fatty acid and glycerol, and you see that the product of the digestion of the fat goes with the name as fatty acid, so this means that they would lower the pH of the solution in which this entire process has taken place. Now, if we just follow the instructions, what they are saying is that these students they added an alkaline solution to the milk, and then afterward they divided the milk into total of four test tubes. Like I would have the test tube, and in that test tube, we would be taking the milk into it, and then along with the milk, the other thing that we are adding here, this is the alkaline solution with us. And after they have done so, so they would take the equal sample of this, and then they would further be taking them into total of four test tubes that we have with us. Now, what they have to say is they added lipase and the bile salts to some of the test tubes, and they have given you the detail of that in table 5.1. So you see, after I have made this solution and I transferred them into total of four test tubes. Now, in these four test tubes, they would be adding lipase into some, and they would be adding bile salts to some. And they further said that they did this at the same time for each test tube. So they have carried the experiment at the same time, and other than that, they are keeping the temperature constant at 40 degrees centigrade. After they have done the protocol, they are leaving the test tubes for the total of 5 minutes, and then afterward, they added the universal indicator solution to each of the test tube, and now they are measuring the color change of that test tube in order to get an idea that whether the fat was broken down into fatty acid and glycerol, and either the content of the test tubes they have become alkaline or they are remaining acidic or a situation like that. So if we just look on toward the table 5.1, here we have test tubes, then we have the contents, and then at the end of that we have got the color of the pH indicator of 5 minutes, and that too for the temperature of 40 degrees centigrade. If I were to talk about test tube A, so they have milk, they have alkaline solution, they have lipase, and they have bile salt. So my idea is that this bile would emulsify the fat in the milk, and the lipase would break down the fat into fatty acid and glycerol, and the orange color would be an indication of an acidic pH. So here we are given the pH ranging from 0 to 14, and the color which they have given you here, so they are indicating that what pH there was in the test tube. So for the test tube A, the color is orange, and this is lying in the pH 2, which is the acidic pH that is indicating that the fats were broken down into fatty acid and glycerol. If we were to go on toward the test tube B, so they are adding milk, they are adding alkaline solution, the bile salts, and the water. Now, the idea here is that we don't have any of the lipase. The bile would emulsify the fat, but since we do not have any of the lipase enzyme here, so it would not be broken down into fatty acid and glycerol. And that is the reason the color here is blue. And this blue is falling in the alkaline range. If I were to go on towards C, here we have milk, we have alkaline solution, the lipase, and the water, and you see that the test tube is showing the yellow color. And if we were to come here, so this again is acidic range, the 4 comes in the acidic range, but comparative to the previous one where we were having this orange color, so this value is little bit higher than 2. So this indicates that here for the test tube C that we have here, the fats were not broken down to a greater extent but to a smaller extent because lipase alone has an ability to break down the fats into fatty acid and glycerol. But without the bile since emulsification has not taken place here. So that is why the breakdown of the fat is not that much efficient. If we go on toward the test tube D, so they are adding milk, the alkaline solution, and the water, and the color again here is blue with us. That again would suggest that none of the fats are broken down. And you see, if we just compare this T to the rest of A, B, and C, so we are neither adding the bile, we are neither adding the lipase enzyme. So this is acting as a control experiment that we include in our investigation to be acting as a control experiment. And this is clearly suggesting the results of the D that this is the presence of the bile salts and the lipase that is responsible for the color change. If we move onward to the first part, they're asking that explain why the test tube D was included in the investigation. And as I have said, that this is acting as a control experiment because neither we have added the bile nor we have added any of the lipase. So this would give us an idea that the difference in the color of the test tube is only when there is presence of the lipase enzyme or the bile salts. If they are not available, then ultimately we cannot have the differences in the color. Now here they are asking for the total of 3 marks that explain why the color in test tube A was orange. So if I just move you back here, this is an orange color for the test tube A because the bile has emulsified the fats and the lipase have broken down the fat in the milk to fatty acid and glycerol, and the presence of the fatty acid chains would make the pH of the solution acidic. So if we just simply read the answer for this, so we would say that the bile in the solution, this has emulsified the fat molecules and they got converted into some small fat droplets. And further, these small fat droplets, due to the presence of the lipase, they were broken down into the fatty acid and glycerol. And once the fatty acid chains, they have come into the solution following the digestion by the lipase enzyme. So this has lowered the pH to 5 and it is indicating that this is acidic pH with us because the orange color that we have here, this is indicating a pH of 2. So this is not 5, we must correct it. Here we are required to be writing pH 2 because that is an acidic pH. Moving further, they are asking for the total of 4 marks that explain the results for the test tube B and the test tube C. So if I just move back to the test tube B and the test tube C, so for the test tube B, the color is blue, and this blue is a kind of an idea that the solution pH is staying alkaline. And the reason is for the B, we have provided the bile salts that has just emulsified, but since we don't have any of the lipase enzyme here, so the fats were not broken into fatty acid and glycerol. So if I just take you to the answer, so we were saying B, there is no chemical digestion of the fat in the milk to either fatty acid and glycerol. The reason was that the lipase was absent, and that is why the color of the test tube that is blue and that have a pH of 8 to 10, which is an alkaline pH, and that is due to the alkaline solution that we have added there. Now they have asked for the test tube C, and if we just move back to the test tube C that we have here, the color is yellow, and this is indicating again the acidic range. Now, since in test tube C, we have added the lipase enzyme but no bile, so emulsification would not happen here. So lipase alone would break down the fat to fatty acid and glycerol, but that breakdown would not be as much rapid as we have seen for the test tube A that we have got here. So if you simply look on to the answer, so we would say that it is yellow color and the pH of this is around 4 to 5, which they've given in the table, and this is suggesting a slightly acidic pH. And this was because we did not added the bile into it. So the fats were not emulsified, and this resulted for the slower breakdown of the fats by the lipase enzyme. Moving onward to this question number two, and this appeared in October, November of 2013, and this was question number two with us. And here they're asking that explain the term balanced diet. Now, why we are associating this question to the biological molecules? Because the diet that we eat or the food that we eat, we take the sugars and the protein, the fats, the vitamins, the minerals, but we have to also make sure that all of these things that we are eating, they need to have a correct proportion of all of these. And why we are required to be eating an equal amount or a correct proportion so that it can provide sufficient energy for the body's metabolism. So my body or your body, it can work efficiently. So for the total of 3 marks, what you would say that the balanced diet simply mean that it contains equal amount or the proportion of the food molecules like fat, sugar, and proteins. This is important that you mention any three of the components that we have in the balanced diet. And further, we would say that this provides sufficient energy for the body's reaction or the metabolism and allowing it to be working efficiently. For the second part, they are asking that state three factors that influence a person's nutritional needs. So you see that the nutritional needs, they depend on what age we are. The older people, their nutritional needs comparative to the youngs or the adults are less. Again, the other factor that we have with us is the gender, and then it would also depend on the activity. The people, those who are very much active, they are athletes or they are playing some games or sport, so they feel more of the hunger comparative to a person who is lazy and who don't work that actively. So we can write the activity. Other factor that we can state here could also be the pregnancy or it could be lactation in case of the female individuals. Now, moving on to word question number three that we have got with us. This happened to appear in October, November of 2016. The question was 4 BC part and the variant was 4.1. And here they have asked that the human they need vitamin D as a part of their diet. And for 1 mark, they are asking that describe a cause of vitamin D deficiency in humans. So mostly vitamin D we get it from the food, or the vitamin D can be made or synthesized in the body when we are exposed to the sunlight. So if the person, if he has less exposure to the sunlight, or if he is not eating more of the fish or yolk or the animal liver in their diet, so that would bring about the deficiency of vitamin D in their body. For the second part, we have got 2 marks, and they say that describe the effect of vitamin D deficiency in humans. Now, the slight idea is that we have got the bones, and these bones they must be strong enough in order to support the weight of my body. So, for strengthening of the bone, we are in the need of the mineral, and that is calcium. So more calcium absorption it would take place in the bones, so more of the bones will be strong and our teeth would be strong. But the slight issue is that this calcium absorption into the bones, this depends on vitamin D. So if the vitamin D is deficient, so this would clearly indicate that our ability to be absorbing the calcium in the bone it would be reduced, and as a result of that you could write that the bones can become soft or the person can develop the rickets or he would feel fatigued all the time. So this way you would score total of 2 marks here. If I were to move further, here they have given you again an investigation kind of a questions, and they say that the scientists have found that the mice can suffer from the type 1 diabetes. And you may know it from your theoretical classes that the person they suffer from type 1 diabetes when their pancreas is not making enough of insulin and they are not able to convert glucose, excessive glucose into the glycogen. So their blood glucose level remains elevated and they lose more of the glucose in their urine as well. So they say that such kind of a situation they found to be happening in the mice. Further, they said that they also found that vitamin D affects the development of type 1 diabetes in mice. Now, their idea is that for sure the mice can suffer from type 1 diabetes, but if they have the deficiency of the vitamin D in their diet, so ultimately this would lead to more development of type 1 diabetes in them. Now, they actually studied two groups of the mice. The first group of the mice, it was normal, and the other groups of the mice, they were vitamin D deficient. and then each of the group of the mice they calculated the percentage of the mice that had developed the type 1 diabetes, and they took the recording every 50 days. Now they have given you table 4.1 to be indicating the results, and if we just look on toward table 4.1, so we have got columns that we need to have an idea of. Here we have got the age and the days of the mice, and then at the top we have got the percentage of the mice that had the type 1 diabetes. Here we have the first group that were the normal mice, and here we have the second group that were vitamin D deficient mice. And if we simply just try to look on toward the table, what we see is that as the days are passing, so the percentage of the mice developing the type 1 diabetes, this is increasing for the normal mice, and this is also increasing for the vitamin D deficient mice, but an increase in the number of the type 1 diabetes cases. This is more for the vitamin D deficient mice group comparative to the normal mice group. But for the initial 50 days, like the number of the cases that we see here, this is same at 0 days. None of the group is developing the diabetes. At 50th day, the number of the cases of the diabetes in both the group is same. And if we further look on toward the days, so when the days they are increasing from 100 to 250 in both of the groups, so the number of the cases for the first group, the diabetic cases are not increasing that much comparative to here where we see a greater increase in the number of the diabetic mice. But another point that I see here is that if we just look on toward day 150, beyond 150, the number of the diabetic cases in the group 2, this is leveling off, like this is remaining same at day 200 and 250. But for the normal mice, the cases have yet not leveled. This means that the leveling of the cases is only we see it for the group 2 rather than for the group 1. Now, this is what they have asked for the total of 3 marks, that we are required to be describing the effect of vitamin D deficiency on the development of type 1 diabetes in the mice, and we are required to be using the data in table 4.1 by which we would support our answer. I have actually described the points, like they are not asking an explanation for an increase in the number of cases, but rather than the description. And whenever the word describe comes in your exam, so you simply have to be quoting him the numeric values with the reference to either the table which they have given you or the graph which they have given you. So you see, I'm just reading these points for you. I have explained it a while ago. So I would say that the number of the cases of the diabetes for the normal and the vitamin deficient mice is same up to 50 days. Like up to 50 days, it's same. Both of them they have got one cases each. Further, I would say that if we go on toward day 100, so at day 100, the number of the diabetes cases are more, and they are more for what group? The vitamin D deficient group, and they are less for the normal mice. And then we have to give him the values. Like for the normal group, the number of cases is 8. For the other group, this is 35, as I have written it over here. And then other point that I see is that the number of the diabetes cases they level off for vitamin D deficient mice early at 150 days. Like if you just look on till 150 days, so here beyond 150 days the number of the cases are leveling off. But for the first group that we have got, which are the normal mice, so the leveling is not happening at 150 days, but rather than it would happen at some later days. So here you see our entire focus was to be writing in terms of numeric values. Now, if we come to question number 4, this happened to appear in October, November of 2016. This was question number 6, and the variant was 4.1. And here this says that we are showing you a DNA molecule, and it has got a total of 2 strands. So you see, I have got this one strand of the DNA, and here we have got the second strand of the DNA. And in one of the strand, they have given you the sequence of the nitrogenous bases, but in the other strand, I have written the letters already, but there were the vacant boxes here. And now they have asked for the total of 2 marks, that we are required to fill the boxes in this provided figure 6.1 to show the letter of the bases on the strand 2 that will pair with the corresponding bases on strand 1. And you know from the theoretical lectures that among the strands of the DNA, we have got the complementary base pairing, and that would mean that if in one strand, if I have got the A nitrogenous base, and in the second strand we would have T. If in the first strand we have got G, so in the second strand we would always have C with us. So such kind of the pairing that we see in the DNA molecules where the A nitrogenous base pairs up with T and the guanine nitrogenous base pairs up with the C. So this is referred to as complementary base pairing. So this is making the things easier now. Against A, I have to write T. Against C, I am required to be writing C. Now here he has given you T, so we would write A here. And here comes the letter C, so we would write up the letter G here. Now, for 1 mark, he says is that state the name for the structure of the DNA molecule as shown in figure 6.1. So we just have to name the structure which is presented here. And this is the typical structure of the DNA molecule that we refer to this as double helix. Now, if we just come to this part, here the things would become little more technical. They say that when molecules of the DNA they are used to classify species, only one of the two DNA strands is sequenced. Now, why we are using the DNA for the classification of the species? Because the closely associated species they have got more similarities in their sequence of the nitrogenous bases. And if there are some of the species that are not closely related to one another, they are distant from one another, so ultimately, they would have more differences among their nitrogenous bases or the nucleotide sequences which they have in their DNA. What they have to say is that first, the DNA sequence from one strand of the DNA molecule from each species is lined up against one strand from another species. So, any of the species that is coming to me in the lab, I would take their DNA out, and then in one of their DNA, I would just sequence it, that what nitrogenous bases or the sequence of the nucleotide they have in them. And then I would place a species B or C next to it, and then I would see that where we have the similarities and where we have the differences. And this is what they are saying, that the bases of the DNA sequence from the same strand can then be compared with each other to look for similarities and the differences. Further, they make it easier. They say figure 6.2 shows a short section from the DNA sequence of 8 plant species. There are 10 differences between species A and species B. These differences are shown in figure 6.2. So if we were to just compare the species, so we have got total of 10 different species of the plants. And here they have given you the sequence of the nitrogenous bases, and they have already done it for the species A and B. The points where we have the differences, like here we have the similarities, they did not highlight it. But wherever there was a difference, so they are highlighting it or they are marking it with the help of the box. Moving onward, for the 2 marks, they are saying that the number of the differences between the DNA sequence of the 8 species, and they have recorded and they have shown you in the table above. Now they want us to count the number of the differences that we have got between the DNA sequences, which are shown in figure 6.2 for the species C and species D, and species G and species H. So the usual idea is that here we have species C and here is its sequence. We have species D and we are given their sequence as well. So we just simply have to see that where the difference actually lies. So I have already done it for you. Here we see no difference, like the sequence of the nitrogenous base is the same, but at this point there is a difference. And at this point there is a difference. For species C this is A, but in D this is coming as C, so this is a difference. And again we have got a difference here and we have got a difference here. So the total number of differences that we have among species C and D, so there are 4 of the total differences. Further, they asked for the species G and H. So again, here we have similarities. Here the entire sequence of the nitrogenous base is the same, but I see a difference here. For species G, this is C, but for H, it is A. So I'm just highlighting it. And the other difference we are seeing here, as here we have G, here is A, so we are just encircling it. Now, if you were to see here, we have got the same sequence, but for the species H, we have got an extra nucleotide which is not there for the species G, so I'm marking it up. So this means that among G and H, the total differences that we have, they are 3 with us. But for the species C and D, the differences that we have, they are 4 with us. Now they want us that we are required to be writing the answer in table 6.1. So the first comparison was done between species C and species D, and we have calculated that the difference among them is 4. And then they asked us to be calculating the differences between species G that we have here and species H that we have here. And as per their arrangement in the table, so we would write the 3 word here. Now, here comes another point. They say that the most closely related species have the fewest differences between their DNA sequence. Now they are asking for the total of 1 mark that state which two plant species which are shown in table 6.1 are most distantly related to each other. And by the word more distantly related to each other, what we are required to be doing is that we will do the comparison of the species among themselves, and we have to pick up the highest number. And the highest number would indicate that they are not closely related, but they are distant from one another. So how you are required to be doing it? Like the species A cannot be compared to species A, their base sequence would be C. But if I compare this species A with the species B, so the differences that we have in them are 10. Likewise, if I compare this A with species C, so again the differences are 10. Now, likewise, if I compare this species D with species A that we have here, so the difference among them is 13. And for the rest of the species, if we just compare them with A, so their differences are shown here. So as I have said, that among the entire table that we are given here, we are required to be picking the biggest number that would indicate that these two species, they are far more distant from one another. And I see that number as being 13. So this means that the species A and D, they are very distantly related. They are not closely related. Now, if we try to be looking on toward the part D that we have with us, so this is more tricky comparative to the next of the parts that we have cracked. And here they say that most closely related species have the shortest distance from the branching point on a classification tree. So here in 6.3, they have made the classification tree and they are saying that the more common branching point we have as it is existing for this species that we have to ace and the species E which they have given us. So this means that among species E and the other species that we have to highlight, there would be least difference among them. And if we just follow this tree, so we have species A here, and the species A is branching from here. And if we follow the tree further, so close to the A species, we have got these species that are listed here. And if we go a little bit more distant away, so these are closely related. But among this tree that we have got, so these two species, they would be more closely associated. Now they say use the information in table 6.1 to complete the classification tree which they have given you in 6.3, and we have to write the letter corresponding to species B, C, D, and G, and we are required to be writing it in the box which is given next to the branch which they have with us. So I am starting my discussion from here. We have got species E. We will directly go to this table that we have with us. Here we have species E, and we have to see that what has got least difference to this species E. And if we just compare this E with the rest of them, so the least number that we have got is 1 with us, and this is coming for the species D. So I would be writing the letter D here. Now we have to crack this one. So this means that the positioning of this species, it lies here. So if these are most closely related owing to their same branching point, so this species would be lying a little bit far away from these two. So this means that the letter which I have chosen for D, I need to pick the next big number. So after 1, the next big number that I have with me is 3, and this goes for the species C that we have got here. So I am clearly going to write the C letter here. Now, if we were to be cracking this figure which they have given us, so here you see that this species H and this which we have to highlight, they have the same branching point. So now I have to compare this H with the remaining ones to see that where we have the least difference among them. So here we have got the H with us. And if we just look on toward the number in terms of the differences, so the minimum number that lies in terms of the difference is 3 with us. So this is coming for the G species. So this means that this H species, this most closely is associated with the species G. So that is why I would write the letter G here. Now we are done with this. Now we have to do something among F and this letter, which we are supposed to be writing it up. Now, if we just follow the letters, we are done writing all of them. So we just have to pick that letter which is missing here. And you see that the letter which is yet not written here, so this is B with us. And if we just look for the B, so here the letter that we have got is 6 with us. So we simply need to be writing the missing letter here. So the B would come here. So this is how you are required to be sorting these branching questions. Okay, so moving on to this question number 5 that we have with us. And this came in year 2016, October, November, the variant was 4.2. And this was question number 1A part. And here they say that milk is sometimes referred to as complete food because it contains all the nutrients that a young mammal requires. So we know it very well that a young mammal is since growing, so it would require a complete proportion of all the nutrients, and thanks partly to the milk it is providing it. And they have given you this table 1.1, and it is showing you three nutrients that are available in the milk, and they want you to answer for the total of 3 marks that we have to be stating one role of each of the nutrient that would be available in the milk and that would be useful for the body of the young mammal. So I have written many points, you have to pick just one. Now here they say the nutrient is protein. So the role in the body is that the protein is one of the fundamental biological molecule that is required for the growth process. This is also needed for the repair of the damaged part of the body like skin, and it is also needed for making new tissues as we have got the formation of the cartilage in our body. So you are required to be writing just one point out of these multiple points that I have given here. So here we have got the lactose, and we know lactose is a disaccharide, and when this would be broken down by the chemical digestion with the help of the lactase enzyme, so the glucose would be released, and that can be used in the respiration, and that can also be providing the energy. And when we talk about calcium, so we know this strengthens the bone, this strengthens the teeth, and calcium is one of the fundamental mineral that is also required for the proper muscle contraction. So you are required to write just one point of out of these points that I have given in the table. If we were to move beyond, the question is from May, June 2017. This was question number 4. The variant was 4.2. They say a balanced diet is required to ensure healthy weight gain as the children grow. So if the children is growing, we need to provide him with the diet, and that diet has to be balanced for 3 marks. They are asking that you are required to be explaining the word balanced diet. So we know it well from the previous question. It came um, previously as well. So we would say that it is a diet that contains equal amount or proportion of the food molecules. And here I have written fat, sugar, and proteins. You can also write mineral vitamins as well. And this would provide sufficient energy that is mandatory for us to be maintaining the body's metabolism or allowing the working going on in our body to be proceeding efficiently. If we come here to question number 7 for the total of 2 marks, they are telling you that the DNA can be extracted from the webs of the spider, and this DNA can be used to identify the species of the spider that made the web and the species of the prey caught in the web. You may have seen in your daily life that the spider guide that we have with us, they are fond of making the webs, and in these webs the spider they would live, but at the same time they would also catch the prey in them and then later would feed on them. Now we are extracting the DNA from the spiders and any of the prey that is caught in their web. Now they want us to explain that how the DNA which they have extracted from the spider webs can be used to identify different species. Now if I were to tell you all of the species they have the cells in them, and in the cells we have the DNA. If we isolate the DNA out and if we try to be looking on toward the double strands that we have in their DNA, so we will find the specific sequence of the nucleotide or the nitrogenous bases like E, T, G, and C. And these sequence of the nitrogenous bases is unique to only one individual species. So we will have some standard readings of these nucleotides with us already, and any of the spider DNA that we are isolating or the prey DNA that we are isolating, so we would just simply see their nucleotide base sequence, nitrogenous base sequence, and then we would compare it to the standard ones. And if we see a lot of similarities among them, so it would highlight that okay, this is a particular species. And if there would be differences, we would say that this is not that species we were looking for. So for 2 marks, what you are required to be writing is that each species have the specific sequence of the nucleotide that lies in their DNA, and it can be compared with the nucleotide sequence of the other species. And if it matches with that species to which we are comparing it, so they are the same species. But if there are differences that we see, then ultimately the species would be different with us. Moving onward, for question number 8, it came in the year 2021. The variant that we had with us, it was 4.2, and the question number was 2 with us. They say the classification of the giant pandas, and they have given you the genus name Ailuropoda, and here they have given you the species name, which is Melanoleuca. They say is debated by many scientists. Like they have started to classify this giant panda, but this is raising a lot of debate among the scientists. Now they have given you this figure 2.1, where this cute panda has been shown to be eating the plant. So this is suggesting that this panda is actually a herbivore with us. And if we were to simply look on toward its ears, so you see it has got the external pinnas and we see the fur on its skin. So this is a confirmation that this is a mammal with us. Now they have given you the further figure of 2.2, and in that they have shown you the red panda and they have shown you the polar bear. But the polar bear in the diagram, this is eating the fish, so this is suggesting this is a carnivore. But if we just look on toward this red panda, the cute guy again here, so this again you see is eating the plant, so this is acting as a herbivore. Now, if we were to move further, they are saying that state one dietary component that is more likely to be found in bamboo plant than in the fish. So you see, the fish mostly is protein based, but the bamboo which these, the giant panda above and the red panda which they have shown here, they are eating it. So it would have more of the dietary fiber, and this would have more of the starch or the sugar content in it. Now, the next B1 part, this comes from the classification scenario. For the total of 2 marks, they are asking you to state two features which are visible in figure 2.1 and figure 2.2 that identifies the three animals as belonging to the same vertebrate group. Among the vertebrate groups, what we have with us, there are mammals, there are birds, there are fish, there are amphibians. So here you see, if we just simply look on them, like all of three, you could say, animals which are given here, so we could clearly see their external pinnas, and their body also has got a covering of the fur on them. So these are two of the major characteristics, the presence of the external ears and the presence of the fur, that would confirm them to be belonging to the same vertebrate group, and they are actually the mammals with us. Now, if we just look on toward this part at which they have given us here, this looks to be very complicated, but this is not complicated at all. You just have to be relaxed and then see what scenario they are presenting. They say DNA can also be used to classify the species, and we have got the molecular biologists, and what they are doing, they are comparing the DNA base sequence of total of 8 species that belong from the same vertebrate group, and they use the differences to draw a classification diagram. Like they came across 8 of different animals like red panda, ferret, giant panda, polar bear, and all of these. And by looking on toward the similarities and the differences which they have in their nucleotides, so they have simply made a kind of a diagram that looks to be looking like a tree. They say this figure 2.3 shows a classification diagram for these 8 species. The shorter the horizontal distance from the two species to the branching point that they share, the more similar their DNA sequences are and more closely the two species are. If I were to summarize this, like if I talk about the red panda and the ferret, so you see that they share the same branching point here. So this is associating, or this is telling us that these two of the species, they are more closely related. And if I talk about this giant panda and the polar bear that we have got, so if we just follow their branching point, so this appears to be similar. So this indicates that the base sequence of the nitrogenous species or the nucleotide is very much similar. So that is why they have got the same branching point for them. Now further they tell us that the scale on figure 2.3 shows the time at which the molecular biologists estimate that each branching point occurred. So if we just follow this diagram further, so they have given you the timeline. And this timeline is indicating, like if I were to talk about the polar bear, so it evolved or it was firstly seen some 20 million years ago. The more we go from here to here, so we are going backward into the history, and if we come from left hand side to the right hand side, so we are coming to the recent time. And this is telling us that at what point each of these different species they existed or they came into the existence. If we were to move further, so this is a technical question for the total of 5 marks. They say morphology can also be used to classify the species, and by the morphology, what we mean is that on the outside, how do the living organisms they appear to look. They say sometimes scientists think that morphology suggests that the giant panda is more closely related to the red panda than it is to the polar bear. Now, if I just take you back, like by morphology, what we mean is that if we simply try to look on this giant panda that we have here and the red panda that we have here, so you simply see like the positioning of the external ears in this giant panda and this red panda that we have beneath, this appears to look same. And other than that, their eating pattern is same. This is also eating the same kind of a plant, and like the herbivore, this is acting as, and the giant panda is also eating the plant. So this is acting as a herbivore. But if we just try to differentiate them from this polar bear, which is eating the fish, so the behavior in term of eating pattern for this polar bear is different. So they say like simply we just look on the morphology of this and this, so they both appear to be similar to one another. If we move further, they say that we are required to discuss the evidence for and against a giant panda being more closely related to the red panda than it is to the polar bear, and we are required to use information that is given in figure 2.1, 2.2, and 2.3 in our answer. Like if we simply look on toward the figure 2.1, they have given you the picture of the giant panda, and the figure 2.2 gives us a picture of red panda, and the third picture that we have here, this is the picture showing us a polar bear. So we simply have to see, or we have to be talking in favor of that these two guys, this and this, they are closely related. But we also have to speak against them. Now, we have to be using this 2.3 as well. So if we just look on toward the kind of a molecular genetics kind of a diagram that we have given here. So this red panda that we have, and this has got more similarities to this animal which is ferret with us because their branching point is same. And if we just talk about the giant panda and the polar bear, so their branching point is common, or this looks to be similar. So this molecular analysis this suggests that this giant panda is more closely related to the polar bear, not to the red panda. The red panda is in terms of the base sequence of the DNA is more similar to the ferret that we have got. So from the figure 2.1 and 2.2, we can be having some points in favor. And from the kind of a tree diagram that is given here in terms of the DNA sequence, so we have got some point to be talking against regarding the statement. So what I am required to be saying in favor is that the positioning of the external ears or the external pinnas for the giant panda and for the red panda that we have here, this appears to be same. So they are closely related and they are distantly related to this polar bear which they have shown you. Other than that, like this is living in the land area, terrestrial area. This also is living in the land area where you see they happen to be feeding on toward the plants, so their eating pattern is same. So this is suggesting close association among them. And in terms of the eating pattern, you see that this would be there somewhere in living in the water areas where they would have a chance to be enjoying a lot of fish which they catch. So in terms of morphology, they look different because you see, like if we look on toward the appearance of or the position of the external pinnas, this is different from this red panda that we have here and the giant panda that we have here. So the in favor the points are coming from figure 2.1 and 2.2, and in terms of the differences, and we have got it in terms of here and here. So this is what I have written it for you for the total of five marks. We would say that the giant panda is more closely related to the red panda because they have the same position of the external ears. And their eating pattern, you see, it is suggesting that they are the herbivores because they're eating grass. And this is something we are getting an idea from figure 2.1 and 2.2. But if I just take you to from the other figure that we have with us, and that was not figure 2.1 or 2.2, but rather than we have this figure 2.3 with us. So this was suggesting more closeness between the giant panda and the polar bear. Why? Because their distance from the branching point is more comparative to the red panda. So this would suggest that there is a lot of similarities that we see between the giant panda and the polar bear, not with the red panda. And the red panda, if we just try to look at when when it did evolve, so if we just follow this, so this was seen some 40 million years ago. But if we see the giant panda and the polar bear, so they are the newer species, and they firstly appeared some 20 million years back. So in terms of this, these two are closely associated in terms of the molecular basis. Red panda is more closely associated or related to ferret, not to the giant panda or the polar bear. And the other hint is in terms of the time frame where they came into existence or when they evolved. Now, for 1 mark, they are saying that state one other type of evidence that is used to classify the species. So you may see my videos on the basis of classification where we see anatomy, we see morphology, and at the end we go on toward the molecular basis. So they have given you the idea of the molecular evidences in terms of the DNA sequences. So other evidence that we can use for the classification is we need to see their internal structures, the anatomy, how come their organs, how come their bones and muscles they are placed within the body, and we also have to cross them together. And if they give birth to an offspring that would be fertile, so this would indicate they are the same species. But if I have got two species, like if I have got the horse and the donkey, if I cross them together, we get mules. Mules cannot reproduce, so I would say the horse and the donkeys, they are the different species, they are not the same species. For question number 9, it appeared in May June of 2021, and the variant was 4.3. And here they say the main component of the milk is a liquid that is an important solvent in the body. So wherever the word important solvent in the body is referred to, so we just need to name it, and this is water with us. Now, moving onward, they have given you multiple columns. Here they have presented you with the nutrient, then we have the elements, and the enzymes are coming here. And at then we have to quote our examiner that what would be the product of digestion. For the proteins that we have, we know it well that they have carbon, hydrogen, oxygen element. In addition to them, nitrogen would be available. If I talk about the enzyme that can break down the protein, so you can generally write it as protease, or the specific enzymes for the protein digestion are the pepsin and the trypsin, and this protein would be broken down into their respective amino acids. If I refer to the fats, so they have three major elements in them. We have carbon, hydrogen, and oxygen. And the fats, they would be digested by the lipase enzymes. And when the lipase is breaking down the fat, so we would get the fatty acid and the glycerol out of it. For the lactose, they have given you the elements, they have given you the product of the digestion. So from the lactose, you need to know the name of the enzyme. You simply have to add A here, and the name of the enzyme would become lactase enzyme. Further, for 3 marks, they are asking that milk also contains vitamins and mineral salts. They want you to explain that why vitamin D is important for the nutrition of the children. You see, the children they are growing, so they need to have some strong bones, and the strong bones can only be existing if there would be absorption of the calcium. And for the absorption of the calcium, we basically require vitamin D. So for 3 marks, how we are required to be answering it, that this vitamin D would bring about the absorption of the calcium into the bones and it would strengthen them. And it is also important for growth, or you could say development, or you could say strengthening of the teeth. And if we know it well that if there is deficiency of the vitamin D, then the young children they suffer from the rickets, where their bones become soft and fragile and they bend under the weight of the body. So 3rd mark you would get when you would say that the vitamin D would prevent the rickets. So the 1st mark for strengthening of the bones, The second mark for the growth, development, or the strengthening of the teeth. And the third mark you would score here when you would say that it help prevents rickets. Moving to this question number 10, it came in May June of 2022. The variant was 43, and this says that the red blood cells contains the protein hemoglobin. For the 2 mark, they want you to state the name of the 4 chemical elements that are found in all the proteins. So the answer is simple. We have carbon, hydrogen, oxygen, and we have the presence of this nitrogen element. For 1 mark, they want you to be stating the role of the hemoglobin. What we know is that hemoglobin is protein. This is packaged within the red blood cells. So either you say that when the red blood cell would be passing through the alveoli, so it would either bind with oxygen gas. If you forget about it, so you could also say that the hemoglobin is a protein that transport oxygen gas around the body. So since they want you to simply state, so the answer shouldn't be that much long. Simply just write it down as it binds with oxygen or it transports oxygen around the body. For this question number 11, that was in the October November of 2022, the variant was 41, and this was question number 4a with us. They say chromosomes, they are made of the DNA. And for the total of 4 marks, the examiner wants you to describe the structure of the DNA molecules. If I were to just simply make a diagram, what we know is that this DNA, it is made up of total of 2 strands, and these 2 strands they wind around each other in the form of a double helix. Say you would score 2 marks, and then you would say that the small repeating units that we have in the DNA, they are the nucleotides, and in the nucleotides we have the nitrogenous bases, like we have A, T, and we have G and C. So these nitrogenous bases they happen to follow the complementary base pairing, where the A would be attracted to T and the guanine nitrogenous bases would be attracted to C, and there would be presence of the hydrogen bonds between them. So this is simply description that we are required to be writing. So what you are required to say or required to write is that this DNA is made up of 2 strands, and these 2 strands they wind around each other in the form of double helix, and we have got the complementary base pairing between the nitrogenous bases. A would pair up with T and G would pair up with C, and for the 4th mark you would say that there exists hydrogen bond between the nitrogenous bases, and that is the reason that since we have got some attractive forces among these nitrogenous bases that are the part of the nucleotide, so our DNA is able to exist in the form of double helix. For question number 12 that we have here, they say that this figure 2.1 is a diagram of the part of the DNA molecule. This came in the May June of 23 and the variant was 42. And they want you to be circling on figure 2.1, which they have given here, where we are required to identify one pair of the bases. So if you follow me, this is one base pair of the nitrogenous bases. This is the second pair. This is the third pair. So for these pairs, you are required to be putting a circle here. So simply I have put a circle here. You can put it anywhere and you would score 1 mark. Now, for the total of 1 mark, they say that the percentage of thymine bases in the DNA of the species is 29%. We are required to calculate the percentage of the bases that would be the base guanine in the DNA of this species. So what I know is that the nitrogenous base A, T, G, and C, their cumulative percentage, this is equivalent to 100%. Whatsoever the percentage we have got it for T, the same would go for A. So what I'm required to be writing here is, or to be coming to the final answer, that the percentage that we have for the thymine is equivalent to the percentage that we would have for the adenine. Now this is 29%, and this would also be equivalent to 29% for the adenine. Now we need to sum them up together and we need to subtract it out of 100 to be getting the total percentage of the guanine and cytosine. And whatever idea is that as much guanine we have, the same would be for the cytosine. So what next I'm required to do is that I have got the total percentage of this. I would subtract 29 percentage of thymine and 29% of the adenine that I have with me. Now the answer that I would get, I simply have to divide it by 2. So that would give me the percentage of the guanine that we are required to be calculating. So if you just follow the point, the percentage of thymine is equivalent to adenine. So this is 29%, 29% for both of these. For calculating it for guanine, I am subtracting these two and then I'm required to divide it by 2. So this would be 100% minus 58%. This gives me 42%, and when I would divide it by 2, so this is 21% for guanine. The same percentage would be there for cytosine as well. Now for 1 mark, what they're asking is that state the name given to the length of the DNA that codes for a protein. So the small functional unit on the DNA that would code for making the protein by the ribosomes. So this is gene with us. Now moving onward for 5 marks, they asked you is that explain how the proteins they are made in a cell. So here you have to be simply following this diagram. If I were to be making an animal cells having a cell membrane in it, and then we have got at the center there would be the nucleus with us, and in the nucleus we have got the DNA, and this DNA is the guy that basically have got the instructions for assembling the amino acids. Now, for instance, if I were to say that these cells require the making of an enzyme which is protein, so the sequence on the DNA that is specific for making the enzyme, it would be copied on a molecule, and that molecule is messenger RNA molecule. This messenger RNA molecule would leave the nucleus, and then it would go toward the membranes of the rough endoplasmic reticulum. And on these rough endoplasmic reticulum, what we have with us, we have the ribosomes. In these ribosomes, the messenger RNA would insert itself. And as per the sequencing or the sequence that we got copied from the DNA on the messenger RNA, the amino acid they would link up with one another with the help of peptide bond. And afterward, when the amino acid would be linked with the help of the peptide bond, we would end up making the protein. So this is the basic protocol for making up of the protein. So for 5 mark, we would simply say that the instructions which are required for the protein synthesis, they are copied on messenger RNA. Later, the messenger RNA, this leaves the nucleus, or you could say this moves towards the cytoplasm. Yeah, this is coming here in the cytoplasm. And then this messenger RNA that we have here, this is inserting itself to the ribosomes. Once it's there in the ribosomes, the amino acid would be brought about by, like there would be blood capillaries from here, the amino acid would be coming inwards, and these amino acid, they would be linked up here at this point with the help of peptide bond, and this process would be technically referred to as the translation process. And we have to be quoting our examiner for 5 marks that the sequence of the amino acid that we are bringing about in the protein, so this is would determine on the instructions that we have got it on the messenger RNA, and messenger RNA you see at the back is taking instructions from the DNA molecule. Now for the total of 2 marks, they say is that DNA controls cell function by controlling the production of proteins. They say state two type of cell membrane proteins. Now, we know it from the active transport that whenever there has to be the movement of the ions from the lower to the higher concentration, so we have got the presence of the carrier proteins. So carrier protein is one of the fundamental protein that we have got in the membranes. The other point is coming from the nutrition in human, where we have got the villi, and in the villi we have got these epithelium of the villi cells, and in these epithelial cells, if I just make the diagram of one of the epithelial cells, so in their cell membrane there is presence of these enzymes, and these enzymes, they also act as proteins. They are protein in nature. So the other word that you could write it over here is that in the epithelium of the villi of the small intestine, we have either of these three enzymes, which are the proteins, like either you could say maltase, lactase, or the sucrase. Moving onward to the February-March of 2024, the variant is 4.2, and they say enzymes are protein. Again, for 2 mark, they want you to be writing the names. You are not required to be writing the formulas of the element that are there in the protein. So this is carbon, hydrogen, oxygen, and then we have this nitrogen with us. For question number 14, again, for the total of 6 marks, they want you to be telling them that how the proteins are made, and as I have said above, for 5 marks you just simply have to write one extra point that you would tell your examiner that the instructions are required for the protein synthesis, and they are present where in the DNA. These instructions, they are copied on the messenger RNA. The messenger RNA then leaves the nucleus and it comes toward the cytoplasm, and then the messenger RNA inserts itself to the ribosomes. Then on the ribosomes again, the amino acid, they are linked by the peptide bond by the translation process. And the last statement is same, that the sequence of the amino acid that we attach within the protein, this would entirely depend upon the instructions that the messenger RNA has brought from the DNA. So I have discussed all of the questions that came from the topic of the biological molecule till 2024. No question came out of this topic in the year 2025. So I hope you would be very much clear about the dimensions of this topic. So I wish you very best of luck.